Today we're going to be continuing to talk about macroecological patterns in nature. As we've already seen, there are broad latitudinal patterns of diversity in fish, in amphibians, particularly frogs, as well as in other groups, including plants. And all of this led us to ask what causes these diversity patterns and why do some areas within the tropics have greater diversity. We also talked about patterns across clades where you get these evolutionary radiations which can be thought of as a summation of patterns across among clades and with biogeographic processes, resulting in these in groups like the Hawaiian honey creepers, bromeliads, Caribbean anoles, and cichlids. We used a broad conceptual framework where we said that individual species geographic ranges are determined by a set of factors. One, they have to arrive at a particular location using some mechanism or the other. They have to stay alive there, encountering both environmental, that is abiotic, as well as biotic factors. And all of this results in where individual species are found. Or in fact you could say where individuals are themselves found. This combination of individuals of a species results in their geographic range, and the summation of geographic range results in species richness. We examined or discussed latitudinal and altitudinal patterns in particular, though you also find these in bathymetric gradients. And so far we discussed environmental factors such as temperature, precipitation, and resources, and we also talked about the relationship between species and area. However, there are other factors as well that have been postulated to influence these large-scale patterns of species richness, and we're going to talk about those today. So coming back to this, on the one hand, you have these patterns of diversity across latitudes, which is from the equator to the poles, both north and south. And you also have a pattern in diversity across elevations, right? And this pattern can be both monotonic decline or it can be a hump-shaped distribution. And there's long been speculation about what causes these hump-shaped distributions. And in the last lecture, we talked about how differences in area at different zones of the mountain might be one of the drivers of this pattern. Now, in the 1980s, George Stevens proposed something called the Rapaport's rule. And he named this after Eduardo Rapaport, who had proposed a similar pattern in mammals, where it was seen that the geographical range size, that is the latitudinal range, the latitudes within which an organism was found, increased as you went from lower to higher latitudes, that is from the equator to the poles. So as you went from lower to higher latitudes, the latitudinal range size itself increased. This led them to postulate this relationship between ranges and latitude as something that was caused by underlying ecological factors, and this relationship was known as Rapaport's rule. Now Stevens proposed something called the climatic stability hypothesis when observing this pattern and naming it as Rapaport's rule. He also said that one of the mechanisms that he postulated might cause this is that temperate species have greater environmental limits. So something that survives in temperate regions, because of the nature of temperate climate, has to be adapted to hot and cold, because there's the annual range in temperature, the daily range in temperature even at certain times of the year were quite substantial. The tropics, on the other hand, experienced both a narrow sort of seasonal and diurnal range in temperature, and also small changes in temperature as you went from one location to another. And so Stevens postulated that this adaptation to those environments and their greater tolerance of a range of environmental factors allowed them to have greater latitudinal ranges as well. In this particular graph, you can actually see that the seasonality of temperature is greater towards the northern latitudes. This is a schematic from the Western Ghats in peninsular India. That the seasonality in the northern Western Ghats is much greater than the seasonality in the southern Western Ghats, which means that any species that is able to tolerate the environment of the north would also be able to tolerate those temperatures in the south. Is this working? Yes. So for example, a species that was able to tolerate these temperatures here would be able to tolerate this range of temperatures, but a species that was only adapted for this might not be able to survive here because its tolerance limit is only from here to here, right? So Stevens's mechanism provided an explanation for why species at higher latitudes might have greater range sizes. Now, Rapaport's rule itself has received a lot of criticism, and there's been some support of it in some instances. There's been many caveats, many counter arguments, and ecologists are divided, or perhaps even lean towards the fact that this is not a universal pattern, and that the mechanisms that underlie it also may not be universally true. However, one important thing that this has led to was, if I go back a slide, when species extend their ranges, so if this northern species had only been found sort of in the northern latitudes, and the southern species had only been species in the middle were adapted here, species in the southern latitudes were here, these would be contributing to the species richness of the regions they were adapted to. But because these have higher tolerance limits and they extend their range all the way south, they overlap in range with the southern species, thereby increasing the species richness here. And this is a consequence of species from the north extending their ranges into the south, and this was one of the outcomes that was proposed, might cause outcomes of these larger ranges of higher latitude species, resulting in the consequence that the lower latitudes had higher species richness. So it was suggested as one possible effect. Now, this led to what was proposed as the mid-domain effect, and what this is a neutral model proposed by Caldwell and Hurt first in 1994, and then Caldwell and Lees sort of presented a more general framework for this in 2000, and they proposed what they called a geometric constraints model, and they said that if you take any bounded domain, as we did in the previous slide, now this could be equator to poles, or it could be the base of the mountain to the top of the mountain, or it could be the any particular geographic range, specifically southern Western Ghats, the tip of the southern Western Ghats to the tip of the northern Western Ghats, any domain that was bounded beyond which species could not disperse because either there was no habitat, or that is bounded by an ocean. So you could think of the continental boundaries of South America or Africa or India, where these are bounded by certain physical barriers. Anything that's a bounded domain, which therefore poses a geometric constraint. And what they proposed was that if you now let's mark it out over here. Let's say I have a bounded domain here. You cannot find any, there's no habitat, or there's ocean here. Now species can only exist between this domain, and they said if I distribute these ranges randomly, so I take species A is found from here to here. What's random about this? And I have obviously just not used a randomization algorithm, but I'm doing it arbitrarily. If I make the range size, so I've arbitrarily taken a range size, I've arbitrarily placed it somewhere else. Then I do the same for species B, smaller but near the left, slightly larger, this is species C, and so on. So let me just do this for lots of different organisms of species for this particular group. Some are very large, some are very small, but the ranges are the sizes of these ranges are randomly selected, and where I'm placing them is also randomly selected. And this was done using a computer algorithm. And what they then did was they said, if I total up now the species found in each of these zones or bins, so now I'm just going to add up the number of species by the number of the total species by the number of species that whose ranges overlap this particular zone, correct? And so for this it would be A, B, whichever one this was, D, E, G, and so on, right? And so on. So I count those up and I say some number, 8, 10, whatever it is. Now, if I draw the ranges in this fashion, what they found is that when the range limits are constrained by boundaries, as we said, you will inevitably get a hump-shaped pattern. So these are actually null models that are gained from derived from an algorithm like this. There's a software that they produced later, which I think there is still a version of online. It's called Range Model, where you can experiment with this and throw 100 species into a bounded domain and see the kind of patterns that emerge from just randomizing range size and position. And when you do that, you get a hump-shaped distribution that looks like this. If range and range size and position are random, you get a more or less uniform hump-shaped distribution, a more smooth hump-shaped distribution. If you use the empirical range sizes, so if you actually now do the randomization, but you take the range sizes from your own data set, then you get patterns that are not as smooth, that are a little bit more influenced by the types of range sizes you have in your empirical data. So Colville and Leese were pointing out that if I now, if I have a mountain range, that if I were to distribute ranges randomly on this by itself would produce a hump-shaped relationship between species richness and elevation. They were similarly arguing that if I look at the globe as a domain, if I look at this as a bounded domain, and I distribute species randomly on Earth, and in fact much of the southern hemisphere doesn't have land, so if I compensated for the amount of land, that I would actually get a hump-shaped pattern through just the random distribution of ranges. And I would get a species richness peak somewhere in the tropics, which is what we see. And so they were arguing, Robert Colville and colleagues were arguing that that if I take biology out of the equation and if I just distribute ranges randomly, this itself can produce the pattern that I often see in nature, and that the hump-shaped pattern on elevations is in fact analogous to the hump-shaped pattern if we look at the pattern on Earth not as being a decline from if I put 0 over here, 0 latitude, people have often thought of it as a decline towards the poles, but if I now look at the South Pole and the North Pole together, that actually this represents a sort of hump-shaped pattern that's also explained to some degree by this null model. Now, this became a very hotly debated controversy in the early 2000s. The question being, how can we just rely on non-biological explanations when we know that biology matters? For 200 years, naturalists and more recently the last 100 years through experiments and observation, ecologists have shown convincingly the relationship between individuals and the environment, populations and the environment, the effect of abiotic and biotic factors on populations and communities. Now, given that we are so keenly aware of this, of the effect that environment and other factors of biology influence species distributions and their survival, how can we arrive at an explanation for a global pattern of diversity across latitudes and elevations based on a non-biological null model itself alone? However, there's an important thing to consider over here, which is what is the basis for comparison? What are we comparing any pattern that we see in nature with, right? So for example, if I were to look at a pattern of diversity, and let's take elevations, let's say from 0 to 3000 meters, and I find that diversity, species richness declines. Right, species richness declines. Now I have some points. Now when you have points and you have a scatter plot and continuous variables on your x and y axes, you would typically do a regression of some sort, a linear or some other. You would fit another model to it. I fitted a linear model here with this trend line, and I would look to see whether I would examine whether that relationship between x and y was significant. Does x, which is elevation or altitude, does x explain the difference in y, the variation in y? Now, if x did not explain the variation in y and my relationship was not significant, I would be, my null model here is that elevation has no effect on species richness. The question that they were asking over here is, should our basis for comparison itself be this uniform model in the absence of biology, given certain range sizes, given boundary constraints? My expectation itself would be this, and therefore I now have to explain this pattern that I see relative to this base distribution and not this base distribution. This was the argument that was being made by Robert Caldwell and others. But as I said, this remained hotly debated. But this had been debated for a long time. When MacArthur and Wilson proposed their theory of island biogeography, where they proposed that the number of species on islands was determined by colonization and extinction, of course, on the premise that all other things were equal. Other ecologists of the time, such as David Lack, strongly argued that factors such as habitat would override these neutral, biology neutral considerations, or these models that were neutral to some aspects of biology. So similarly, there was a long-standing debate between Robert Caldwell et al. About the role of the mid-domain effect versus other factors, particularly environmental factors, and with people, for ecologists like Michael Rosenzweig, who argued that area has sort of a preeminent effect versus many other theories. Much of the underlying sentiment in this, as I mentioned, was that we know that environment is important. Also, early on, there was criticism of some of the papers because the statistical methods that we used to demonstrate the role of the mid-domain effect was flawed. When you compared, say, environment, when you drew a graph and said the correlation between temperature and species richness is that when temperature goes up, species richness goes up, and then you draw another graph and say that the predictions of the mid-domain effect and species richness are well correlated. It was pointed out that temperature is a predictor. My results of my model of the mid-domain effect is a prediction, and this is not a very statistically rigorous way to examine this question. Some of the early work that sort of showed that these may not act in isolation included a study on Western Himalayan plants, where the first pattern the first result was that when you actually look across a number of mountain ranges, the individual patterns, sorry, let me go back a second, you might find, especially when you get a combination of species with different adaptations, such as you typically would along a gradient. In this case, let's say tropical and temperate. The tropical species richness declines with altitude. The temperate species richness increases with altitude. And because the middle elevations have a greater species pool, when you actually sum across a large number of individual gradients, you end up getting a hump-shaped pattern, even though the ecological relationship with the environmental factor is monotonic, is unidirectional, and so you get a decline with temperature for tropical and an increase with temperature for temperate species. So the relationship is actually linear, but when you sum these across a large area, you might still end up getting a mid-domain effect. So the mid-domain effect not as a consequence of overlap, but as a consequence of species pools in areas where different species with different biogeographic affinities are overlapping. Interestingly enough, it doesn't matter what the pattern at the base level is. So maybe they don't respond to temperature. You get a mid-domain effect with both tropical and temperate species. That's the base pattern. Biology is not determining this. But when you sum across many different individual ranges within a mountain range such as the Himalayas, you end up getting the same mid-domain effect. So the mid-domain effect at the larger spatial scale does not necessarily imply mechanism at a smaller spatial scale. And finally, one of the first examples of combining temperature and environment, where tropical species with small ranges are sensitive to temperature, decline with elevation. Temperate species have broad environmental tolerances and therefore follow what you would expect, therefore, from a classical mid-domain system where species have broad tolerances, have moderate to large ranges, lots of effective overlap, and therefore show a hump-shaped pattern. You combine these two as well, and you still get at the largest scale, you get a sort of mid-domain effect, but perhaps a little bit skewed towards the left, towards the southern elevations, which is actually the pattern that you see in nature over there. Now, having said that, models that actually effectively combined range and environment did not in the early 2000s exist. And so a group of ecologists came up with this idea of range cohesion and range scatter. And the idea inherent here is simply that the underlying factor that results in the overlap is the fact that these ranges are contiguous. And so they came up with a set of models that predicted the distribution of organisms based on the ranges are contiguous, as one would expect from dispersal, and that they are not. And now within range cohesion and range scatter, you could assign occurrence probabilities based on environmental parameters. Now, just to be clear, you remember I said what's our underlying assumption? If ranges are scattered, so let's go back to how ranges are distributed. Now, ranges I'm going to draw range cohesion below and range scatter above. So range cohesion is what I showed you. Each range, that is the species, is found in all the locations from here to here, here to here. Each one line is a different species. They're found in all these below, right? These are contiguous ranges. Now let me take ranges of the same size. So let me take A. If they were scattered, what that would mean is that the range total size is the same, but they are no longer contiguous. Now I take species B and I do the same thing, slightly smaller range, species CDE, and even smaller range. Now maybe it's distributed like that. Now if my ranges are contiguous, then I would expect that mid-domain pattern or the hump-shaped distribution. If they are scattered, regardless of the distribution of the size of ranges or the location, I will actually get that uniform pattern that we were originally comparing our statistical models to. So range scatter produces this uniform pattern, range cohesion produces this hump-shaped pattern, and allows us therefore to compare the effect of environment against both null models, null model 1 and null model 2, right? And this is the one that historically we had always been doing without realizing. When we drew a set of scatter points on a graph and drew a regression line, let's say again this is temperature and species richness, and drew a scatter plot, and then drew a trend line, calculated the R square value, looked at the significance of that R square value of the regression of the F statistic in this case. Now what we were doing was effectively, the null model here is that there is no effect of our x, variable x on y. Effectively, therefore, we are comparing it with this model, and this model is represented in our modeling domain now as the range scatter model, as these models where ranges are not contiguous in the way that they are here, right? So now this actually gives us multiple models as baselines against which to compare whether environment has an effect or not. This was first presented in a paper by Rapaport et al. 2007. To give you an idea of how it works, the cohesion model, essentially they modeled organisms as moving through space in a contiguous fashion, and this could happen in two ways. One is it could randomly move, as I just showed, but you could also predicate, or you could also say that the next cell it moves to depends on the environment being suitable over there. So this could be a pure range cohesion model with no environmental effects, or a range cohesion with some environmental affinity built into it, right? Now similarly, we could build a range scatter model where the next cell is completely random, it's randomly picked out of a hat, or the next cell it goes to is where the environment is preferable. So that would be a range scatter model, but determined entirely by the environment. Now this is sort of like an everything is everywhere, and environment determines, filters what you find there eventually and not. Go back one more step to these, as you can see the difference is that because of cohesion, an organism that starts in the middle does not have the same access to all of the cells in the domain in the sense that the range scatter models do. It has to move by this process of contiguous dispersal. So if it had started, for example, in this cell over here, it would have access to these cells to begin with, and it would probably be less likely to reach here depending on its dispersal ability, right? Now in scatter, any cell in that domain is potentially occupied by an organism, and only the environment determines whether it's found there or not. So Rapaport et al. Applied this in 2007 to South American birds, where they applied both range and cohesion and scatter, and they built a bunch of models with and without environment, including energy, water, composite variables, and so on, and did show that range cohesion models did show with moderately sized ranges did better than range scatter models, and that water energy variables were also important in determining the distribution patterns or the species richness patterns in South America for these birds. Now, of course, this also depends on dispersal ability. So one way to think about it is that different taxa are affected to a different degree by environment. They're also affected to a different degree by dispersal. And so species that have small dispersal abilities, on the average, let's say the very small dispersal abilities like many frogs, may have very tiny ranges, and range overlap may not play much of a role. On the other hand, birds generally disperse large ranges. Many birds have large distances. Many species in a group have large ranges, and you would expect the effect of range overlap to be greater in a group such as birds. Finally, we come to evolutionary factors, and some of the interesting patterns that have been that have been seen in nature, and the underlying processes are explained by hypotheses such as the phylogenetic niche conservatism hypothesis that was proposed a couple of decades ago. This talks about the inability of a clade to invade and diversify in novel environments. So the phylogenetic niche is conserved. All the descendants of a particular clade have similar niches. And in particular, this has been used to explain tropical conservatism. Warmer temperatures and abundant rainfall characterizes the ancestral niche of many species. One particular example contrast comes from amphibians, where in the entire group of anurans of frogs and toads, you see a clear decline in species richness from tropical to temperate latitudes. In fact, beyond a certain latitude, you get very, very, very few species. On the other hand, as you go towards temperate latitudes, the richness of newts and salamanders, which is another group of amphibians, increases. So the ancestral niche of newts and salamanders was colder environments, whereas the ancestral niche of frogs and toads was warmer and wetter environments. Other things have been proposed. Evolutionary time. So richness has been related to the time available for species to evolve and fill empty niches and habitats. Also evolutionary factors that we'll talk about a little bit more in the biogeography module. Repeated upheavals from glaciations and climatic drying, and as a result of this, repeated extinctions and less time to diversify. So to sum this up, let's come back to that question: why do some areas within the tropics have greater diversity? And I want to add now evolution to what we discussed before. So there's species geographic range, which is how individuals, why are individuals found at the locations we find them, right? And the sum total of individuals at particular locations within a species gives rise to its geographic range. We've so far talking about ecological factors. We said it had to disperse or get there somehow. We talked about the biotic and abiotic factors involved in staying alive. But if we start at the beginning, we can say that the two reasons that can explain why an organism is found at a location. One is that it started out there, so it evolved there, it speciated there, or that it dispersed there either in ecological or evolutionary time. Once it's there, it has to stay alive, it has to persist, and that persistence is mediated by its own traits, but it has to deal with both biotic factors such as competition, predation, parasitism, and so on, and a whole host of abiotic or environmental factors. And the sum total of many geographical ranges gives rise to these species richness patterns. And so these are the broad frameworks that we use, both evolutionary, biogeographic and dispersal, evolutionary biogeographic dispersal and ecological, in order to get a better understanding of species diversity and distributions. Going forward, on that particular note, the models that combine range and environment and evolution now, actually I should say plus evolution, are of interest. These provide that broader set of mechanisms that will allow us to understand these patterns. As Gotelli et al. Put it in their 2009 paper, we can think of these as three knobs for predicting species richness. The knobs being dispersal, evolutionary origins, and environmental gradients. And by knobs, what they mean is that, like with any knob, you can sort of like dial it up or dial it down, and dialing these knobs up or down actually results in the patterns that we see for different taxa. So for example, the knob for dispersal is probably dialed up for birds, while the knob for evolutionary origins might be dialed up more, in the sense that it plays a greater influence in explaining patterns in frogs. Overall, we combine all of these as we did here in this framework to understand these broader patterns of diversity and distribution. Thank you.